Okay, for real. Thanks everybody for joining us for the OpenWorm Journal Club. Today we're talking about how does a worm crawl, the neurobiology of locomotion. Review of two papers, one by Cohen and Sanders and the other by Georgieva et al. From this year, 2014. And joining us today we have Cohen and Sanders, who are joining us here from the University of Leeds. Would you guys like to introduce yourselves?
Hi, I'm Netta Cohen. I'm interested in dynamics of complex systems, and I've been interested in this little nematode C. elegans for the last 10 years or so. And I've, in particular, been interested in understanding how the locomotion system works, thinking initially that it's probably very, very simple. And I've learned to be surprised by it and love it.
And I think the reviews that we've got here are going to show just why it's not as simple, but why it is so interesting. Tom?
Yeah, I'm Tom Sanders. I'm a PhD student in Netherlands lab, third year. I work on reflexes, intelligence, sensory integration, plasticity, and
Great. Okay, excellent. And joining us here in the room to ask questions during this presentation and to sort of have a discussion going, we've got several folks joining us, various community members from around the OpenWorm community. So let's have you guys introduce yourselves. Let's start with Johannes.
Yeah, okay. So hi everyone. My name is Johannes. I'm actually studying physics in Germany. And yeah, I'm currently taking part in the Google Summer Code program at OpenWorm and working on the simulation environment.
Great. And Matteo?
Hi, I'm Matteo Cantarelli. I've been working on the OpenWorm project for the past 3 years, I believe. I'm an electrical engineer and I do software mainly. I worked on the OpenWorm browser, and now I'm working mainly on Geppetto, the simulation environment Johannes was referring to.
Great. Board?
Hi, I'm Boris Gleason in University College London. I'm a postdoc, and I've been involved in OpenWorm for a few years, sort of various different areas, but mainly involved in the expression of the nervous system and trying to get that into NeuronL, which is a modeling language that I'm also working on, and try to integrate that into, hopefully, into the smooth particle physics simulation eventually.
And the other Tom.
Hi, my name is Tom Portuguese, and I've kind of been with the project for almost a year now. My background is in machine learning, and so I'm trying to apply some of those techniques to train the connectome to do some behaviors.
Awesome. Terrific. And I'm Stephen. I'm a coordinator for the OpenWorm project, and I'll be presenting slides today. As I mentioned, we've got two papers, and one is authored here by our guests, Netta and Tom, and so I've put together my best sort of reenactment of that, plus with content from the other paper, plus with content from some of the references and just some other details to kind of fill in for a bit of a broader audience. So, but I've asked Netta and Tom to jump in if there's anything that needs to be augmented or added to, and I will also probably toss out some questions for them as well. So as to kind of keep a good discussion going here. So without further ado, let's go ahead and go on to the presentation. Okay. So these are the two papers. I have posted the link to these slides on the Hangout, and there was technical difficulty, so you may need to go from one Google Plus event page to the one that we're on right now in order to get those links, but please have a look at those. So these are the two papers, and they've come out very recently, just in the first half of 2014. And they both talk about the question of how does the C. elegans, this little nematode, create locomotion? How does it create the behavior that causes it to move? And this is a question dear to our hearts at the OpenWorm project, because we are interested and excited and engaged in creating simulations of just such a thing. And this is an example of the beginnings of an approximation of what we've got that does some kind of crawling. But at the moment, this model purely uses a sinusoidal input, and it doesn't yet draw from the extensive neuronal model that we built and that the literature reports. And so we are reading these papers with a lot of interest to kind of understand and learn not only some of the best examples, but the whole landscape of what is known right now about the neurobiology of what makes this kind of behavior happen. So, the Georgieva 2014 paper had a nice thing at the beginning that I kind of wanted to start with, which was the way that systems are a proposed means of breaking down systems analysis that was proposed by David Marr, who was originally a computer scientist, but also did work in vision and made contributions to many different areas back in 1982, and he proposed this three-part split of analyzing systems that the Georgieva paper used as a way to describe their investigation, and I thought was nice for this too. So there's a computational way to look at a system, which refers to its inputs and outputs. Then at the bottom, there's an implementation way to look at a system, which is what are the components of that system and how are they connected. And then in the middle, there's a space for the algorithmic, which is how is the computation at the top implemented by the components and their connections at the bottom. And this kind of breaks down quite nicely into three areas as it relates to the C. elegans and its behavior, which is that the computational could be viewed as its behavior, meaning that it receives inputs from the world and it moves in a certain way. The implementation is sort of its anatomy and how are neurons in its body connected and how are its parts connected together. And then the algorithmic section is sort of the information processing. How is that behavior implemented by the anatomy? And kind of being able to focus on all three, I think, is helpful to guide our conversation as we go forward here. So starting then with the top, with the behavior, so both papers describe the behavior. It's important to note that the behavior of C. elegans, in a very gross sense, can be thought of as crawling and swimming, whereby in a swimming case here up in the upper left, the worm uses essentially a full wavelength bends in order to propel itself, whereas in a crawling behavior, it uses wavelengths that are much shorter than its whole body to move itself forward. And we pulled a few movies of what this looks like. So this is actually from the worm behavior database, which is cited here. And this is an example of a worm crawling through an auger environment that is fairly viscous. And in contrast, the swimming behavior is shown here. Oops. In this movie, where you can see how it's rather different. The worm is bending quite a bit more, and you can see that big wavelength that's talked about there. So now the Cohen and Sanders paper points out that until recently, these behaviors were considered to be distinct gates that the animal switches between in response to the environment. But as they point out, with quite a bit of citations and evidence there, that these two behaviors are now being thought more like extremes of a continuous spectrum. And that's important because it suggests a mechanism that a single computational model should implement that can account for both. And so this is something in recent years that has been an insight into these behaviors that suggests a particular way of looking at the problem. Okay, so as we're going to move into some of the connections, for everybody who's coming into at a very basic level, I just want to remind everybody about synapses. So a synapse is a connection between a neuron, between neuron A and neuron B, and we either think of them as chemical synapses, which is a particular kind that has neurotransmitter, or we think of it as a gap junction, which is more like an electrical connection. Under the microscope, these look very different. A chemical synapse has, this is sort of a slice through a cell where there's a cell connected at the top and a cell at the bottom. Okay, so we call this a presynaptic area and a postsynaptic area. And neurotransmitter is held in vesicles in the presynaptic area and spills across a gap here and is received by receptors in the postsynaptic area. Whereas a gap junction is an electrical synapse, and again, a sort of a side view slice. These are more viewed as pores that allow the electrical potential of one cell to be shared with the electrical potential of the other. So we'll be talking about synapses here in a moment, so that's just kind of important to remember. And also, another important bit of nomenclature for everybody to remember is we'll use the words dorsal and ventral quite a bit. And if you're not up on a lingo, right? So dorsal means sort of the back side of the top, if you will, the top or the back, I guess. If you were to put it upright, this would be sort of the back. So along the place where the spine is, if you think about a shark and its dorsal fin, it sits along sort of the back of its body. The ventral is the bottom or the belly of the worm in this way, and then anterior is up towards the front and the nose, and posterior is towards the back. And as you can see with this particular worm, its bend here is in a dorsal ventral axis. In fact, that's how all of the crawling or swimming happens with the worm, is it's creating dorsal ventral bends. So when we say that dorsal ventral thing, don't be confused, that means that we're creating bends that go from this back axis to this belly axis in the way that's sort of shown here. And in fact, when it's crawling, it's sort of sitting on its side, so it doesn't actually wiggle in the other axis. It wiggles in this particular axis here. Okay, so the Cohen and Sanders paper starts out, okay, now kind of combining these ideas together, now that you understand dorsal ventral and you understand, synapses, sort of points out at the highest level here, a simple way to think about what's going on when those dorsal ventral bends are being created is that there's there's 4 major types of motor neurons that we're going to pick here just to simplify things that are responsible. So DB and VB are cholinergic. In this case, that means that they're excitatory. So there's kinds of cells that excite other cells. And in this case, they excite both muscles on either side of the body, as well as they excite neurons that are inhibitory. And those are the other 2 kinds, which are the DD and the VD neurons. Now, I know that these may be quickly confusing, so let me just, as a clue, the first letter of these neuron names, the D and the V, refer to dorsal and ventral. And the last letter refer to sort of a type of the neuron. So you can think of these 2 as the B-type motor neurons, and these 2 as the D, as in dog, the D-type neurons. And so then what we're talking about here are the dorsal and the ventral B-type neurons, and the dorsal and the ventral D-type neurons. The B-type neurons are excitatory. They're the cholinergic ones. And the D-type neurons are GABAergic or inhibitory. So at a very basic level, it's important to understand that what motor neurons do is that they connect to muscle cells, and they excite them, or they inhibit them. And what we're saying here is that in forward locomotion, the basic thing that happens is that cholinergic motor neurons are exciting the muscles either on the dorsal side or the ventral side, and then they inhibit the opposite side that they aren't exciting at that time by exciting GABAergic inhibitory motor neurons. Netta, how am I doing?
Great.
Yep.
Okay.
Very clear. Yep.
Okay. All right. Very good. So now I actually wanted to take advantage of the fact that we have some 3-dimensional views of what these types of neurons that I just went over look like for everybody's benefit here. So this is just a quick sort of 3-minute movie. Maybe I'll skip around a little bit. So these are what the VB neurons look like, the B-type neurons that are excitatory that are on the ventral side. And you can see, as I said about ventral, that's along the belly of the worm, which you can see there. And this is known as the ventral cord, actually. This whole system that runs along the body is the ventral cord. And that's sort of a key anatomical landmark for producing these behaviors. So these are the excitatory guys, okay? Now, if we, let's see, so if we, and what basically, oh, that's right. And the other thing is that they're numbered from 1 to some number. It depends on the class that you're talking about. There's more or less depending on the type. And they're ordered from the posterior to the anterior, well, in reverse, I guess. They go from the highest number is in the anterior and the lowest number, the highest number is in the posterior and the lowest number is in the anterior. So VB1 or VV1 or VD1 are in the front up here, and they go backwards the other way. So, okay. So those were the VB neurons, right? And then if we skip ahead, we get to the DB neurons, which are the excitatory ones, and as well, and they cross over from the ventral cord onto the dorsal side as well, and they are excitatory. If we go a little bit further here in this, we'll get to the VD neurons. So these are the GABAergic neurons that are inhibitory. And you can see that they're also crossing over and sort of following a fairly similar pattern, but still distinct. And then, if we go a little bit further down here, we'll get to the DD neurons, which are GABAergic and inhibitory. So what is often going to be summarized here in a schematic, like we were seeing before, as basically circles, right? So going back here. Right, so in this schematic here, we've taken this side view of the worm, and you notice now this ventral nerve cord, which you were seeing in 3 dimensions, is highlighted in red. And in this box, what we've done is we've basically taken a portion out of this, and we're looking at just basically a little cross-section of the worm, and we're looking at basically in schematic the relationship between a given DB neuron and a given VB neuron and a given DD neuron and a VD neuron, sort of in an abstract view. But what they're doing is they're stimulating those various muscles. I also wanted to point out in this movie as well, I think I had the muscle view in there towards the ends. Where'd the muscle view go? Okay, yes, here. So here you actually see the muscle cells themselves. So there's the neuron. If I wait a second, then, okay, so this is what the muscle cells look like in 3D, and there are 4 quadrants that run along the body of the worm in this way, and these are the muscles that are being stimulated by the neurons that we're looking at. And here I'm sort of making that more transparent, so you can see how they run along, how the ventral cord here runs along the bottom. But basically their target are these green cells that are around the edges there. Okay, so just a little bit of a lay of the land of what that's looking like. All right, now switching over to the Georgieva paper, which also starts to break down some of the anatomy, now the picture is a little bit more complex. So in fact, if you only had in your picture, if you only have DD and VD and DB and VB, there's actually more to the story, but it's a useful place to start, basically, with those 4 sets. So so here we add in a little bit more description, but essentially these are also other classes of motor neurons that are involved in the circuit. And to give you a flavor of what that looks like, without getting too deep into the complexities of this network, you can kind of see some of these other neurons starting to fit into this picture. When you look at this table that came out of the Georgieva supplemental, that started to talk about the relationships between our 4 classes that we started with here, which again, so in this column they talk about the neuron to muscle connection, so you can see that the VD and the VBs are the cholinergics, and they connect to ventral and dorsal muscles. Again, VD and DD are GABAergic, and they connect to ventral and dorsal. They connect to other motor neurons themselves, and so other guys like VAs are fitting in here, and AS are coming into the picture as well. And then they also get input from other interneurons that are involved in the picture, like PVC, AVB, PVN. So of course, this is a whole network, and there's lots of neurons in it. So to kind of start to tease this apart, sometimes it's useful to have a simplification. Do you want to say anything as well that might be like useful rules of thumb to understand the roles of some of these other guys that are layered in here, Netta, or maybe just sort of say, like why you did start with this, with just these 4 to begin with?
I can say a couple of things. Perhaps first of all, a caveat about worm wiring.
Yep.
So when we look at the circuit of the worm, it's a model.
Yep.
Models are approximations and are based on either our best estimates of the data, which themselves are approximations, or simplifications and abstractions of that. The specific schematic that we're showing here, which is sort of leading up to a specific model, or even to a class of models, is basically saying, let's limit only to those classes of neurons that have been directly implicated in forward locomotion by laser ablation studies. And yes, we can say a few words about what those other classes of neurons are. Altogether, there are 8 classes of motor neurons in the ventral nerve cord.
Right.
But beyond that, it's simplifying the wiring of those 4 classes of motor neurons. So, because what we were ultimately aimed at is a computational model, and a computational model that somehow captures the essence of what's required to achieve this forward locomotion, rather than exactly mimicking the worm, then it helps to have fewer parameters and fewer unknowns in your model, and therefore simplifying the circuit is a very powerful tool. So we followed in the footsteps of others, in particular of Ernst Niebuhr and Paul Erdos, who already in the 1990s suggested the simplification of repeated units of neural circuitry. And these repeated units allow you to simplify the system so that the number of motor neurons of each class is the same. Rather than having to deal with how do you connect 7 of these, 11 of those, 13 of those, and 6 of those.
Yes.
So
Exactly.
that certainly was a huge simplification, allows us to basically endow all the neurons with similar properties, and is a huge simplification in the model. It is at the same time a caveat that obviously the reasons for the worm having different numbers of neurons, and hence more complex connectivity, is not known to us. And what the advantages and limitations of that are in the actual biological system is not known to us. And that remains outside the scope of many of these studies that we've done to date. We don't really understand what's going on there.
Exactly.
If we sort of look at the overview of what we think these neurons are doing, we have the VBs and the DBs. So the B-class motor neurons are the primary motor neurons that have been implicated in driving forward locomotion. And those seem to be assisted by these inhibitory motor neurons, the Ds, as you suggested. And we seem to have something very similar for backward locomotion, driven by excitatory motor neurons called A, again, dorsal A and ventral A, except that those ventral and dorsal A's connect to the shared inhibitory neurons, the D class. And in addition to that, we have these AS's, which stand for A-like but short, because their axons are shorter. And we don't exactly know what they're doing, but actually there's some really interesting insight from recent papers just looking at the connectivity and saying, well, maybe from the connectivity we can glean some insight into what they might be participating in. Possibly backward locomotion, possibly coordinating between forward and backward. We don't really know. And in addition to that, we've got the ventral C-class motor neurons, and those are really divided into anterior, middle of the body, sort of around the vulva, and a few at the posterior end. The ones around the vulva are have been implicated actually directly in egg laying. And so we've got the VC 1, 2, 3, and 6, if I remember correctly, which are probably implicated in locomotion. And the closest hint we have of that is by a series of papers that implicate them in the ventral bending during omega turns.
Cool. Awesome. It's tempting to try and draw parallels between that B and D go together and then A and C go together because of their names, of course. But that's probably an oversimplification as well. But
yeah.
Yeah.
The D class are shared between the forward and backward circuit. So it's B goes together with D, but A also goes together with D.
Yes.
Okay.
Share neurons.
Okay. All right. Very good. That's very helpful. All right. So some of the this is just sort of giving some of the sense of the connectivity here and some of the major players. But thank you, Netta, that for illustrating that this is a model. These are simplifications. It's always important to keep that in mind. Okay. So I wanted to get into sort of the meat of this sort of three hypotheses of locomotion that the Georgieva paper puts out, and then I think that in the context of the Cohen-Sanders paper, a lot of the discussion kind of fits, I think, into the third of the three hypotheses. But I wanted to give the first two some additional commentary and probably also get Netta's perspectives on that. So I am going to put on the parking lot, though, a couple things that were in the Cohen and Sanders paper just for now, because there's quite a bit of good stuff to get into. Maybe come back to them towards the end, which there's some comments about navigation, which I thought was really interesting, actually, about how sensory neurons, there's evidence that they themselves do nonlinear processing as independent units, which is pretty neat, and don't require like a circuit necessarily to do some processing. And also, and some of the advantages of that from an engineering perspective, that having your nonlinear system up at the sensory boundary can be very helpful, and as well as then having nonlinear stuff sort of at the outside for output. But I don't want to go too much into that. And then neuromodulators, of course, is quite a bit of time spent in that paper talking about neuromodulators, which, of course, it's important to recognize that they really kind of cross-cut and really complicate this whole story that we're going to be talking about with neurons and circuits that are purely neuronally driven. There's a lot of evidence that can in fact reverse the flow of information, whereas normally we think things go from sensory to interneuron to motor neuron. What's pointed out here in this review is that in fact motor neurons can release peptides that affect sensory neurons. So it throws a whole other wrench in the story. But for the moment, I'm going to leave these aside because the hypotheses that the Georgieva paper goes into doesn't really touch on either of these topics. Okay, so then another vocabulary word, just if you're joining us from outside of the neurobiology sphere, a word that's important is proprioception, which is the perception of stimuli relating to position, posture, equilibrium, or internal condition. This is a general Merriam-Webster definition, so it's not worm specific. In our case, proprioception is really most related to this internal condition idea, the idea that the nervous system is aware of itself, of its own body, I should say, that the worm is aware of its own body and its own position or its own posture, its own internal state. So that's going to be a key concept here that we're going to get into in some of this theory. The other key concept that I wanted to introduce here, because it's also pretty important, is that of the central pattern generator, because it's kind of a straw man that's been a really important thing in C. elegans, locomotion. And I wanted to give Eve Martyr the opportunity to introduce it just very briefly here, because she's actually the I believe credited with being on the paper that sort of really pushed forward central pattern generators as an idea, and it is a very important topic in neuroscience. So just a couple minutes from Eve talking about what a central pattern generator is.
Two central pattern generating circuits, or CPGs as we call. Now, rhythmic movements such as walking or walking sideways or walking sideways in the other direction are produced by central oscillatory circuits, in this case in my spinal cord, but for many animals in various and sundry ensembles. And these circuits have been studied, or central pattern generating circuits have been studied in a variety of animals, including leeches, lampreys, flies, snails, frogs, mice, and chicks. But one of the most useful preparations has historically been the crustacean stomatogastric nervous system.
Okay, and that crustacean stomatogastric nervous system might not roll off the tongue, but is actually a very important circuit in its own right, and really the subject of Eve's great work over the last many years. Okay, but the important thing is I've introduced that as kind of a foil, because you notice that in that list of things that it's been studied in, it's not been studied in C. elegans. And in fact, the search for a central pattern generator, a central pattern generating circuit in the C. elegans has so far gone, has gone vacant. And this quote from Cohen and Sanders sort of point that out. By entirely lacking a central pattern generating CPG circuit along the body, these models differ substantially from other central peripheral pattern generating mechanisms, from leech, swimming, through insect stepping to human walking. So now I've excerpted a little bit here, and some of the things I've left out is that they point out that there are in fact models that propose some kind of a central pattern generator, even though there is not yet sort of the biological evidence for it. And Georgieva will sort of talk about that. In fact, their first hypothesis they put out involves a central pattern generator that's in the head and the neck. And I actually wanted now to throw this over to Netta to sort of if you can, did I get that right? Is it that these things presume a CPG, but that it hasn't been found, or would you say it differently, or is that incorrect?
Okay, so I'll say a few things. First of all, that quote from the previous page was referring purely to models, not to the worm.
Okay.
I don't think anybody would say in so many words that the worm lacks a CPG.
Okay.
You know, we haven't found it yet, but in locomotion. But it doesn't mean it's not there.
Okay.
There are clearly oscillating neurons in C. elegans. So, for instance, defecation is an example of where there's probably going to be something rhythmic which is endogenously generated. And almost everybody thinks that there must be some sort of central pattern generated control in the head circuit. And there have been some suggestions as to what these might be, but not detailed models per se. The question that we dealt with is what happens along the body. And the question that the Georgieva paper deals with is a broader question of, is there a CPG altogether? So there may be or may not be a CPG in the head. I would say probably there is. And the question then is, if you stop the head from doing whatever it's doing, would the body be able to still oscillate or not? Does it require this head? And
right.
So if there is central pattern generated control in the body is one hypothesis. If it's only in the head is another hypothesis. If it's in both, then that raises all kinds of interesting questions, right? So the first hypothesis is, as I see it here, is that there is a CPG in the head or some oscillator in the head, and through whatever mechanism, this is able to propagate down through the neural circuit all the way down the body. And then the questions are, first of all, what generates the oscillations in the head? They could be a CPG, or they could themselves be proprioceptively driven, or some combination. And then what drives this? How does this propagate along the body?
Exactly.
And that takes you back to the question of, is there a CPG along the body, or is there just proprioception along the body, or is there some combination?
That's right. That's right. So the Georgieva paper was trying to make these distinctions between models, which probably are more different than they are similar, to sort of group them together. I believe that this one, some of the evidence or some of the proposals had been for specific neurons, for example, AVA, that might be responsible for doing the propagation, but that there had been evidence that had been found that that AVA is not active in correlation with forward locomotion, and so that had cast some doubt on the idea that propagation is an essential component. Is that about right?
Right. So historically, people were thinking about these things that in C. elegans nomenclature are called command interneurons. AVA for backward locomotion, AVB for forward locomotion, and a bunch of others, as basically on-off neurons. They're on when they're giving a certain command, they're off otherwise. But there hasn't been electrophysiology of these neurons during real behavior. And recently, it's finally been possible to do some calcium imaging on them in behaving worms. It's not electrophysiology during real behavior, but it's close. It's as close as we can get in C. elegans.
Right.
And so yes, these papers that have done electrophysiology, sorry, not calcium imaging in freely behaving worms seem to indicate that AVA and AVB are indeed command neurons.
Yeah, so they don't oscillate on the time scale of undulations. They also don't really show an on-off pattern. So it's not that they're on during forward locomotion or on during backward locomotion. They show something a bit more complex. But it does seem like they are command interneurons involved in either the initiation of forward or backward locomotion or the maintenance separately.
Right. Right. Okay, excellent. So then just so everybody sort of understands what this model is, sort of, because it's sort of setting up a pattern here. So what this is saying is that in the head of the neck, an oscillation is occurring. That oscillation is then transferred by some means down to the rest of the body. The purple arrows are showing that this is that sensory feedback is involved in this process. The pink little cylinders here are the muscle cells, is sort of the schematic for muscles as they would go down the body in this way. And so this is just showing the muscles sort of in flat here, and this is showing them in sort of the curved model of the worm. Okay, so that's hypothesis 1 from the Georgieva paper. Hypothesis 2 from the Georgieva paper suggests, looked at some models that suggest that, in fact, there are coupled oscillators that exist throughout the ventral cord, that are each themselves capable of producing a rhythm independently of each other in this sort of segmented pattern that I think Netta was alluding to earlier, that it's useful to think about these potentially independent units to some extent down the body of the worm, and then sort of locomotion is happening because these guys sort of sync up and are oscillating sort of out of phase with each other, I guess, in order to create the forward motion. And this then doesn't just rely, in contrast to this, doesn't just rely on propagation, but each of these themselves are essentially a generator of a rhythm. And both papers pointed to this idea of this other paper, Haspel and O'Donovan. Haspel is, I believe, also a co-author on the Georgieva paper, to kind of back this up. And this idea that there are independent units that are themselves capable of generating oscillations. And I will let Netta here at the end correct me if I misstated anything. But my understanding of what this idea of the perimotor space is to basically align the motor neurons, the classes of motor neurons here, along an axis from the head to the tail that corresponds to their connectivity with muscle cells, such that if you have, so these circles along the way here with numbers in them indicate the numbers of the cell that falls along this horizontal axis. So this 3 here means VD3 or VD8 or VD12, right? So this is an individual neuron, and its position along the horizontal axis has been picked based on its connectivity to the muscle cells that are directly beneath it here. And the dotted lines here are showing you, for every cell, it's showing you which are the muscle cells that it's mainly synapsing with, or that it's mostly influencing or affecting. And so the idea of the perimotor space is just first of all, just to lay these guys out in this sort of ordering so that you can kind of give a coordinate to each neuron that corresponds to either its type on the y-axis or its position along the head and tail on its x-axis. And if you do that, this is sort of a setup. Okay, so what does that tell you? What is that worth? So if you do that, what this paper proposed and what the 2 review papers have cited is this idea that that in fact you get some nice grouping of repeated segments. Repeated segments is proposed to be 6 of them along the length of the worm that can be then composed in a single segment. So if you think of, there's one segment more anterior and one more posterior, that's kind of a combination of the types of motor neurons here that can be connected together in a pattern like this. and this is what it would look like in the 6 perimotor segments as you go along the body of the worm. So when I saw this, I don't know if I had described this well, Netta, but it did kind of look. This is purely driven by an anatomical, an anatomical analysis of what could be possible. I don't know that this has been validated functionally yet, but it looked like this was an appealing way of sort of breaking up the motor system. Can you say a little bit more though about how you see this?
Yeah. So yeah, you're absolutely right. So this is not data. This is interpretation of the data. It's another model in a sense, in the sense that it's a schematic and an interpretation of where to align the neurons along the body in a way that's consistent or most consistent with a known anatomy, based on these electromicrograph sections. Hopefully, when we get more EM data or more annotation of the EM data, some of this can be checked at the anatomical level. There are some simplifications that were required to even get this 6-unit repeating motif.
Right.
Nonetheless, there does seem to be some evidence that there is something like that. But then the curious question is, if we just say, look, let's go along with this. We don't know if it's right or not, but let's go along with it. That's the purpose of models, is to drive your curiosity and get you to explore things. And so if we go along with this, one could imagine that these units could be responsible either for propagating a signal from the head or for generating a signal as a single unit or in a distributed way across the different repeating units. So it's not necessarily saying that these are going to be, each one of them, a pattern generator. If we look in other animals, for instance, in the lamprey, then where they've got hundreds of segments, it's not clear that a single segment can act as an oscillator. Sometimes it takes several segments to act as an oscillator, and we have no idea what's happening in the worm. But the ability to find repeating motifs is a nice step forward, because clearly the simplification of 10 or 12 or 100 repeating units or 13 repeating units based on the number of a particular class of neurons, as was done by Niebuhr-Nerdos and then by some of our papers and by others, is clearly just a simplification, whereas this is taking a step closer to the anatomy.
Yep. So very good. Yeah. So it's important to point out that, yeah, just because you see a pattern doesn't necessarily mean that they would be these oscillators either. They could just as easily be, the means of propagating this sort of signal. So it doesn't lend credence to one or the other. I sort of put it under here, though, I guess just because it was an appealing place to put it, to kind of think about these idea of a circuit that is repeated. And I decided to go a little bit further as well because we can, and just kind of say, like, well, what does this look like in 3D if we picked one of the proposed perimotor segments, which also involves some muscle? And I don't know that I got a ton of insight out of it, but I decided to have a look at it anyway. And it's sort of a group of this is now a cross-cutting look, not just at any one specific class of neuron, but looking at all of the neurons that would be proposed to be in one of these repeating units, at least in one of the simplifications that was on the paper. So it's kind of interesting. All right, now hypothesis 3, and this is the one with the Georgieva paper really cites quite a bit of Netta's work over the last several years, so it's really great to have her here. So this one suggests that there's not an oscillator per se in the way that we had been thinking about it before, but that environmental neuronal interactions is what gives rise to the locomotion behavior, and so they pointed to the 2012 work with Boyle and Cohen that generated a model that did not require a head, that only had sort of just the segments beneath the head, and it was able to, with a single parameter, smoothly transition between crawling behavior and swimming behavior. all using the same mechanisms, which is something I don't believe that any of the other models had been able to do. That was an extra feature of your model. Is that right, Netta?
Yep. So, yeah. So the model doesn't say anything about the head.
Right.
It basically is a model of the body.
Right.
And saying, had there not been a head, or had the head been disabled, could we get undulations? So, in a sense, it's a thin line between hypothesis 1 and hypothesis 3.
Okay.
Hypothesis 1 also mentions, if you look back at that slide, it also mentions sensory feedback along the body for propagating the signal.
Right. That's right. That's right.
This
Yep.
case, assuming that that sensory feedback mechanism is not generative in itself, it's only propagating. But that's a thin line.
Yep.
Between propagating and generating.
That's right. There's a lot of sort of ambiguous causality here, right? So I sort of read this one to sort of say that the head was both was necessary, as in there couldn't be locomotion if there were not an oscillator. Whereas I viewed hypothesis 3 as saying that it's not necessary, and in fact you don't have to have it at all. But of course, as you're pointing out, the picture can be much more complex, and causality in these sorts of systems are often tangled. So this is the I called it no oscillator model in more detail. I'm not sure if that's the best way to describe it, but sort of quoting from the paper here in the second figure. So the idea, so I thought this formulation was actually rather nice, and just kind of to walk through what this picture is. So this is sort of taking the DB and the VB neurons, which we know from before are the excitatory neurons, together, and sort of saying in this simplified state diagram, let's think of them either as on or off, and if they're off, if they're not exciting, then they're a 0, and if they are exciting, then they're a 1. And we start assuming perhaps some kind of a top-down input that kicks this process off, maybe not. But either way, this process is kicked off. And so we start then from this picture of a dorsally bent configuration. So in this case, the two lines here, just to orient yourself again, these are sort of another schematic of the layer of muscles on the dorsal side, and a layer of muscles on the ventral side. And so we're starting with this bent dorsally, so bent more towards the back of the worm. And so they're initially off. So initially both the DB and the VB are not exciting anything. And then sort of saying stretch input on the ventral side, okay, will activate VB. So this idea that the ventral side is actually has motor neurons that potentially themselves can detect and do this process of proprioception, which I was talking about before, or by some means anyway, those neurons become active based on the fact that the ventral side is stretched. And so then when VB is activated, then ventral bending is gradually going to increase stretch on the dorsal side, which then eventually activates DB, and we get down to here in the 1-1 state, which then automatically resets VB via the inhibition, so we get over here to the 1-0 state, and then dorsal bending continues until the stretch input, the dorsal side falls below threshold, releasing VB from inhibition. So the idea is that it's this cycle that is causing the locomotion, that there's not per se a neuronally based oscillator, but an oscillation occurs as a result of going through this loop. Do you want to add anything to that, Netta?
So yeah, no, that's a pretty nice summary of it. I think I might add that a key criterion for you to believe this schematic is that it should be able to work no matter what the initial shape of the worm is.
Okay.
So if you have to bend it ventrally for this to work, or if you have to start from state 0-0 or from any particular place that's exactly along this cycle for it to work, and it's not going to be robust to changes in conformation, then you've got a problem.
Right.
So indeed, this sort of schematic does show that even if you start the worm from a completely flat configuration, There's going to be some asymmetry in how the worm responds, how the ventral side responds, and how the dorsal side responds, so that some side has to turn on. In this case, the ventral side will turn on if the worm is completely flat. But the key point I think that's important to note is that you can't possibly understand what's going on here without the embodiment.
Right.
So if you didn't have these pictures of which way is stretched where, you wouldn't be able to understand anything. And that's quite a different way of understanding neural circuits from what's conventional. So if you've got a central pattern generator and you're generating an oscillator using the neural circuit, you don't need to worry about what the body's doing. You look at the neural circuit, you see what the neural dynamics are, then those drive the muscles, you look at what the muscles do, then those drive the body, there's some interaction with the environment, there's some forces, there's some biomechanics, and fine. But in this picture, you can't look at it in a sort of feed-forward way like that. You need to look at the loop with the environment at every point in time.
That's right. And I kind of wish now that I would have included a slide on this idea of the stretch receptors that are, I think, proposed in the VB motor neurons. Is that right? Can you say a little bit about those?
So we don't know where stretch receptors are. Our best guess is that they're either in B motor neurons, class B motor neurons, or in muscles, or in both. There was a recent paper by Wen et al., not so recent now already, a couple of years, that gave some evidence that in support of stretch receptors in the B motor neurons, but we haven't seen it conclusively yet in terms of the molecular details of how this works and where this works and whether this also exists in muscles or not and so on. So there's a lot of work to be done in terms of elucidating what the proprioception is. In terms of the B-class motor neurons, this was first postulated already in the 70s. People looked at the anatomy of the B-class motor neurons based on the earliest EM micrographs and saw that they had these really long posterior extensions that they call axons. And in the what's known as the proximal part of these axons, in other words, the part that's most anterior, so these axons are facing the tail, bizarrely enough. So you've got a cell body called the soma, and then you've got this long connection towards the tail, where this is the head of the worm. And in the area that's relatively anterior, that's where you have your neuromuscular junctions and all the rest of it, and you can see why that's functional. But in the distal ends, nobody could see any anatomical significance to having these extensions so long. And so that's why people postulated already back then that they might be important for proprioception, that they might be sensing the shape of the body. Whether that's true or not, we don't know.
Yeah, very good. Well, it is a fun feature, certainly for folks coming from other areas of neuroscience, to think about the neurons themselves being the cells that can sense the shape of the body, if it's possible. You know, certainly there are ion channels that we know are capable of that, but often they're on sensory neurons, and so I think that's it's a very intriguing thing, and one of the reasons why the neuromechanical models that you've been engaged in building and we've been engaged in building are interesting, and I think as you point out here, if this model holds, then it's actually crucial, and that you can't actually know what the nervous system is doing without it. Okay, so I just wanted then to, I know we're getting close to time here, and I want to at least have 10 minutes to open up discussion. But just as a way of summarizing, I just want to go back to MAR again and just kind of review what we talked about. So we showed the movies of the behavior. We talked about swimming and crawling. We talked about how a lot of the evidence is pointing at this being not being separate gates and running with separate systems, but in fact all being driven by the same motor system. And that's kind of the computation level of analysis. We looked quite a bit at the anatomy, and we looked at the circuits, and we talked about the classes of motor neurons that are involved, and the simplified version and the more complex version. We also looked at this prospect of repeated patterns and some anatomy that might back up segments that are independent, or at least can do things on their own potentially. And then we went through the three hypotheses from the Georgieva paper which really focused on how do those two things come together? How do you get the behavior that we're seeing there from the anatomy, from the muscles and the motor neurons that we're seeing there? And obviously, what both reviews point out is that we don't have the final answer yet, and that there's still more work to be done in order to kind of get to additional conclusions. And so the jury is still out. But what I was excited about reading both these is how much we do know, and how much additional knowledge I needed to fill in for myself in order to get a better sense of kind of what the lay of the land looked like. So I'm very happy for these summaries, and I'm really glad that we got a chance to go through them. Okay, so let's break there for a moment. And so we've been pretty quiet, guys, here in the room. I haven't heard anybody sort of speaking up, so I'm hoping that you've been assembling lists of questions diligently, and I can bring up any slide that you want to talk about as well. But you guys have any questions for Netta and Tom? Please, somebody come off.
I had a question.
Great.
And this is I have sort of a selfish reason for asking this, but at the end of your paper you mentioned about the peptide chemical signaling mechanisms that Stephen also mentioned. Is it possible in some way that those are involved in learning? And my selfish motive is that in artificial neural networks, there's a mechanism for backpropagation of signals that don't seem to have any basis in biology. So here we have something that maybe, I don't know. Do you think that's there's a learning aspect of here with those peptide signaling things?
That's a big one. If I might, I wouldn't say that there's no evidence whatsoever for backpropagation. So there are backpropagating action potentials. There are some indications that there's stuff going on in the brain, though it's not exactly what backpropagating feed-forward neural networks do, or not even close to that. But with respect to what neuromodulators do and neuropeptides do in particular, they change the system. I don't know where to draw the line between changing the system and learning. So a standard definition of learning is that it is a change in behavior that is based on experience. Now, if based on experience, so your behavior is driving the release of some neuropeptides, which is causing your behavior to change, then that qualifies as learning. If you mean, do neuropeptides cause memory consolidation in the sense of synaptic modifications, then there's some evidence that C. elegans can do that. But again, I don't know whether you want to call that modification of behavior or learning. But yes, neuropeptides affect cell dynamics, but they also seem to affect neurotransmitter release. They modify neural circuits, and so if any modification of a neural circuit based on experience qualifies as learning, then yes. Whether this is something that you can go and extract and apply to the human brain is too much of a leap. But if I didn't think that there was some mileage in that question and that it deserves exploration, I wouldn't be interested in C. elegans.
Well, does C. elegans exhibit any sort of what you would call memory consolidation, or is it just too simple to do that? Because I'm fine with just, weight modifications, but...
Right. C. elegans does not have a hippocampus.
Right.
It does not have a thalamus. It does not have a cortex. So a lot of those terms that come from vertebrate systems and from mammalian systems and even from primate systems are very loaded by some understanding of a brain architecture.
Yeah, but you would think some feedback would be necessary.
You have some memory that lasts hours. You have some memory that can last up to 24 hours. We don't understand well enough which of those memories are cell autonomous, which of those memories are synaptic, which are a combination. A lot of the learning seems to be in the sensory layer, possibly even in sensory neurons, and some seem to be in synapses that are directly downstream of sensory neurons. And so a lot of the questions have to be sort of phrased very carefully.
Fair enough.
For many, many hours.
Okay.
Yeah, I think it's a difficult story really to look at learning in C. elegans for many reasons. One is that, of course, these neurons have graded potentials, but also graded synaptic release. So if you think about your typical long-term potentiation and long-term depression, where you look at the correlation of activity between a presynaptic and postsynaptic neuron, that doesn't really work when you have constant synaptic release. So it's not that like you have it in mammals, the presynaptic cell fires, that causes the postsynaptic cell to fire. Okay, you know, that's correlation, so the connection strengthens. While in C. elegans, there's a constant release. So it's, yeah, these things don't work, and I think that's also why things like LTP and LTD don't really seem to exist in C. elegans.
That's a really good point. All right, panelists, other questions?
The other thing that doesn't exist in C. elegans is Dale's law.
Is the what?
Dale's law, if you've heard of that. So that's the idea that a single neuron is either purely excitatory or purely inhibitory. So it has only one neurotransmitter.
Yeah.
That doesn't hold in C. elegans.
Yeah. Also, the research into into neuropeptides is pretty much just starting. It's really been ignored, or it wasn't deemed to play a big role. But it seems now that also in mammals it could play a big role, and you know the research is just starting. So really we don't know whether in humans it could play a role in learning. But like Netta said, it's interesting, and it's interesting. Sure.
Awesome. Matteo, you look like you got a question.
Well, I don't think it's a proper question, but I always try to think about the future. And I think I asked Netta this kind of questions before in the past, but like looking forward in terms of like obviously this is an ongoing effort that has been ongoing for years. It's like new models, new hypotheses, test them, see how they do. And now we had recently the whole animal calcium imaging that was done, and there was a paper on that. So I'm kind of curious in what way that will influence the future, and in what kind of ways we could help, really, in terms of post-processing the output, for instance, of whole animal imaging in the C. elegans, in order to think and build the next model that we could use in order to test this kind of hypothesis and see how the locomotion works. Like, how could we help, and what would, like, the one or two things that you would say, if we had that right now, that would be very helpful, because that would enable us to do A, B, and C, and we were waiting to do this for years.
So ultimately, the dream in C. elegans is to have a full understanding of the whole animal. That's the dream in C. elegans in terms of genetics, in terms of development, and also in terms of the neural control. And this is something that you can't really dream about even for vertebrates. But when you have hundreds of neurons, and you have the anatomy to such a high level of accuracy, and you're beginning to have the technology to image the dynamics that's going on this circuit, then you can think about doing that in C. elegans. You have to be careful that you can't do the whole animal without knowing the details. And you can't do the details and forget about the whole animal. And so we're constantly going back and forth between sort of a reductionist attitude where we say, let's look in detail at this neuron, or at this molecular pathway, or at this neuropeptide, or at this subcircuit, and saying, well, how does this interface with everything else? And that's also why we're looking sometimes at neural circuits in isolation, and then sometimes saying, no, 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 we need to look at it in a behaving animal with a body, with neuromechanics, with a physical environment. And we go back and forth between these different levels of integration. What I think that is really exciting about whole animal, whole neural system modeling is that it will enable us for the first time to begin to glimpse at how different subcircuits interact during behavior. So yes, it will give us insight into what this neuron is doing during this particular behavior. But what's much more exciting is that you can then start correlating and say, when the worm is performing forward locomotion, this other circuit is doing this. How do we actually understand the integration and the interfacing between different neural circuits? And if you look back at this, at the three hypotheses that were given in the Gervina, Haspel, and Byron paper, which we touched on, not in those words and not in that sort of format in our review as well. There are hypotheses about what's going on in the head. There are hypotheses about what's going on in the body. And because we have no idea how to interface them, they're sort of hypotheses. If we had the interfaces, if we truly understood how the body would work, that would impose very strong constraints on how it must interface to the head and what the head must be doing. And if we understood what the head was doing, this would impose very strict constraints on how this must be interfaced to the body. And so if we can crack some of those interfaces and look at the correlations between subcircuits and see to what extent they coordinate among themselves and to what extent one subcircuit controls the other or is controlled by it, or in a distributed system, in a synergistic way, generate the behavior, I think this will be very, very exciting for trying to move forward towards a whole animal model.
That's interesting. Thank you.
Okay, guys, so we are at the top of the hour, and I want to hold people time. I want to really express so much gratitude to Netta and Tom Sanders for joining us here and being our expert and holding my feet to the fire and keeping me honest about these points. And obviously, just sharing your great knowledge on the subject. Thank you so much. To our panelists, Tom, Porig, Matteo, and Johannes, for being here and having good questions and just sort of. I think that we're all going to take something away from this and think about it further. Obviously, more questions will be generated from this. And for folks watching, hope you enjoyed it as well. This will all be posted up on YouTube afterwards, so you can, if anything went by a little too quickly, you can always get back to it. So thanks everybody very much for your time, and look forward to more OpenWorm Journal Clubs in the future. Thank you.
Okay, thanks very much.
Thanks.
Thanks.
Bye.
Guys.
Bye.